Welcome to A-Level Biology. If you look at past papers for Cambridge 9700, you will see the temperature rate graph absolutely everywhere. It is the backbone of Paper 2 structured explanations. It is a classic Paper 1 multiple choice graph, and it dictates exactly how you design water bath experiments in Paper 3. Today, we are going to master exactly why this graph curves up gradually, peaks, and then drops off a cliff. It is all about a delicate balance between kinetic energy and structural stability. Let's break it down.
Let's start by looking at temperatures below the optimum. In an aqueous solution, enzymes and substrates are in continuous random motion. For a reaction to happen, they have to physically collide at the right angle and with enough energy. At low temperatures, molecules have very low kinetic energy. They move sluggishly, meaning collisions are rare and often lack the energy to initiate a reaction. When we increase the temperature, we add heat energy, which converts into kinetic energy. The molecules move much faster. This has two critical effects. First, it increases the overall collision frequency. Second, it means a higher proportion of those collisions possess the required activation energy. The result? More enzyme-substrate complexes form every single second. This creates an exponential curve upwards. You must use this exact phrasing in the exam to get the marks. You state that more kinetic energy leads to more successful collisions, which means more enzyme-substrate complexes form per unit time. You do not just say they collide more. You must specify that the collisions are successful and explicitly name the enzyme-substrate complex. Examiners are actively looking for these exact terms.
This exponential rise isn't random. Below the optimum, it follows a strict mathematical rule. This rule is measured by the temperature coefficient, known as Q10. The Q10 value tells us the factor by which the rate of reaction increases for every 10 degrees Celsius rise in temperature. For most biological enzymes between 0 degrees and the optimum, the Q10 is approximately 2. This means the rate of reaction roughly doubles for every 10-degree increase. This doubling effect is exactly why the initial slope of our temperature rate graph curves upwards exponentially. Let's look at a classic calculation. First, always calculate the actual rates. At 15 degrees, 4 milligrams divided by 10 minutes gives 0.40 milligrams per minute. At 25 degrees, 8.4 divided by 10 gives 0.84 milligrams per minute. Now we apply our formula, putting the rate at the higher temperature on top. This gives us a Q10 of 2.1. As expected, it is approximately 2, meaning the rate just about doubled.
As temperature continues to rise, we eventually hit the peak. This is the optimum temperature. It is the exact point where molecules have the absolute maximum kinetic energy possible without disrupting the enzyme's delicate three-dimensional structure. The frequency of successful collisions and the rate of enzyme-substrate complex formation are at their absolute highest. For most human enzymes, this sits perfectly between 37 and 40 degrees Celsius, matching our normal body temperature. But this optimum is a delicate balancing act. Even a slight increase beyond this point will cause the rate to plummet.
Pushing past the optimum temperature triggers a catastrophic structural failure. If we heat the enzyme above its optimum, the rate doesn't just level off, it plummets rapidly to 0. This is denaturation, and you must describe it as a precise sequence of events. As the temperature rises, the heat energy causes the atoms within the enzyme molecule itself to vibrate violently. This violent internal vibration breaks the relatively weak bonds holding the tertiary structure together, specifically the hydrogen bonds and ionic bonds. Because the tertiary structure is unraveling, the precise three-dimensional shape of the active site is lost. It distorts. It is no longer complementary to the substrate. Therefore, no enzyme-substrate complexes can form, and the reaction stops completely. This process is almost always irreversible. Pause the video and try this. A student writes, at 60 degrees, the enzyme dies because its peptide bonds break. What are the two biological errors here? Enzymes are non-living proteins, so they denature, they don't die, and heat breaks the weaker hydrogen and ionic bonds of the tertiary structure, not the strong covalent peptide bonds of the primary structure.
While human enzymes peak around 37 degrees, enzymes in other organisms have evolved as adaptations to extreme environments. Psychrophiles live in freezing environments, like Arctic sea ice, with optima around 5 to 15 degrees. Because environmental kinetic energy is so low, their enzymes have highly flexible tertiary structures with fewer weak bonds, making it easy for substrates to bind. The trade-off is they are highly unstable and denature easily at room temperature. On the other extreme, thermophiles live in boiling hot springs with optima around 70 to 80 degrees. To survive massive thermal vibration without denaturing, their enzymes have highly stable tertiary structures packed with extra hydrogen bonds and strong covalent disulfide bonds. This locks the active site firmly in place.
Let's pull everything together. Here is exactly what you need for the exam. Below the optimum, you must talk about increased kinetic energy leading to more successful collisions and more enzyme-substrate complexes. Above the optimum, you must state that thermal vibration breaks hydrogen and ionic bonds, the tertiary structure is lost, the active site distorts, and it is no longer complementary. And never, ever write that an enzyme dies or that peptide bonds break. You are now ready to tackle any temperature question Cambridge throws at you.